Hello, welcome. Today we are going to talk about pre-training and representation learning from proteins. Proteins are biology's actuators. Almost everything that needs to be done in a living cell is done by a protein. For example, human cells contain between 1 and 3 billion proteins each, and they're involved in structure, metabolism, and signaling. IgG is a protein involved in the adaptive immune system in recognizing invaders to the body. Hemoglobin carries oxygen in red blood cells. Insulin regulates blood sugar levels. And adenylate kinase and glutamine synthetase are both enzymes involved in core metabolism. An enzyme is a protein that catalyzes a chemical reaction. Actin is a structural protein. It's also involved in muscle contractions. And firefly luciferase is what makes firefly buds glow. All of this diversity in structure and function comes from just 20 molecular building blocks called amino acids. Every amino acid has a COOH group, a carboxylic acid group on one end, and the other end it has an H2N, an amine group, hence the name amino acid. And then the other part is the variable part, is the side chain. For example, cysteine has a sulfur and hydrogen over here. Threonine has a methyl group and a hydroxide group, and so on. And all that diversity between amino acids comes from the different chemical and physical properties of their side chains. And during protein synthesis, the protein is made one amino acid at a time in the ribosome. It comes out of the ribosome one amino acid at a time, and that's the primary structure, and that folds into things like sheets and helices, and we call that secondary structure, and then that folds into a three-dimensional shape, and we call it tertiary structure. And for most proteins, that's the functional form. Why should we design proteins? There's a bunch of reasons. Some of them include we might want to design new chemistries that aren't seen in nature, but that proteins can catalyze. For example, the Arnold lab a few years back convinced a protein to make carbon-silicon bonds, which are not seen in nature. They might be good therapeutics. For example, asparaginase is an enzyme that breaks down the amino acid asparagine, and it turns out that certain cancers can't make their own asparagine. So if you dose a patient with asparaginase as part of chemotherapy, it kills off the cancer cells faster than it kills off healthy cells. Maybe we want to learn how they work. For example, there's been a lot of work recently on trying to figure out how the coronavirus, and especially the coronavirus spike protein, works. And sometimes we're interested in proteins as molecular tools to study other things. And the most famous one is green fluorescent protein, or GFP, which is small and glows bright green, and we can stick it places to see what to use it as a molecular label. So how do we design proteins? Well, we know that the primary amino acid structure or sequence, which we can represent here using a string of letters where each letter represents an amino acid, completely determines the structure, which completely determines the function. And there's question marks there because we don't necessarily know how sequence determines structure or how structure determines function. But the design problem is really which sequence will give the desired function. So we're going to start from the function and go backwards to the sequence. And that's even harder. It turns out if you sidestep that problem and just do directed evolution and make random changes, you can win a Nobel Prize. Machine learning is one way we can optimize protein properties with fewer measurements by taking full advantage of the information we gain from every measurement. For example, we could start with one or more parent proteins, use some sort of mutagenesis strategy to create a variant pool, then we'll screen that variant pool and get a bunch of sequence function pairs. We use that to train a machine learning sequence function model, and then we use that model to guide further rounds of experiments. For example, there's a study that I did in my PhD with a bunch of other people, where we optimized the ability of this protein called counterrhodopsin, which sits in cell membranes and is light sensitive. And when you activate it with the right wavelength of light, it opens up and allows sodium ions in. And our goal here was to induce as strong of a current as possible. And we managed to do that in about 200 measurements. More recently, there's another paper where they were able to increase the ability of an enzyme called acyl ACP reductase to do fatty alcohol synthesis by about threefold in less than 100 measurements. Let's dive into that counterrhodopsin study just a little bit more. So here we start with 3 parents, and we measure their photocurrent, which remember is their ability to open up and allow sodium ions into the cell in response to a light activation. Then we design a library that consists of blocks of sequence from those parents, chopped up and shuffled, and we synthesize all the single block swaps from that possible library. And most of those are actually worse than the parents. And you'll notice that there's actually a bunch that aren't shown because they either aren't made by the cell or don't even make it to the membrane, so we can't measure their photocurrent. But there are a few that do better than parents. If we allow more diversity and sample for as much diversity as possible for training, most of those, even if they're correctly localized in the membrane, have no photocurrent, although there is one that is remarkably lucky and good. So now we train the model, and over 2 rounds of model training and then selection, we're able to create sets of counterrhodopsins that contain some of the strongest photocurrents ever observed in the counterrhodopsin in a neuron. And we were able to use that to control mouse behavior in the mice that have been genetically engineered to express these proteins in their brains, in certain parts of their brains. We can control their behavior with light without having to remove their skulls. So that's pretty cool. While experimental measurements of a desired function can be slow and expensive, we do have large sequence structure and functional databases of proteins. And we're going to talk about how we might use something called pre-training or representation learning to leverage the information contained in those databases. So the best known and best curated protein sequence database is called UniProt. It contains about 250 million protein sequences, and most of these are found through genome mining. If we deduplicate identical sequences, we're left with UniRef 100. It's about 236 million sequences. If we then cluster them to 90% similarity, you're left with 116 million sequences, or to 50% similarity, you have 42 million sequences in UniRef 50. Now, most of these sequences have never actually been observed as a protein by a human. We just think based on the DNA sequence that there's a protein coding region there. However, a subset of these, about 250,000 in SwissProt, have been experimentally verified. And of those, we have experimental structures, either EM or crystallography, for about 169,000 of those in the protein data bank, in the PDB. And all those numbers are a lot bigger than the numbers we saw, the 200, 100 in the last 2 machine learning guided protein engineering studies. So how exactly does pre-training work? The idea is to transfer knowledge from one task to another. So during the pre-training stage, we take information from our database and use that to train a machine learning model. I'm showing that it's training by turning it a different color. Then we take that model and we fine-tune it on those very expensive measurements from the library we care about. And then we can take that model and make predictions of new sequences that it hasn't seen before and use that to decide what sequences to make next. Many pre-training methods pretend that proteins are language. So here's this very small short protein. So I've written it down as a sequence of amino acids. And that's a little bit analogous to like tokens in language, like letters or characters or words or subwords. So maybe we can use tools originally developed for language on these amino acid sequences. For example, in language, there's what's called a distributional hypothesis, which is that words with similar meanings occur in similar contexts. We can take our protein sequence and chop it into, say, k-mers. In this case, we'll do 3-mers. And this method is called word-to-vec. Chop it up, and now I'm going to take every 3-mer or every k-mer of whatever length I want and encode it as a vector or a list of numbers, and in this case, I'm going to take a window of 3 and use the k-mers in the sides to predict the k-mer in the middle. And this learned representation, that those vectors, those lists of numbers, we're going to call an embedding. And the idea here is that that embedding will represent and somehow encode the physical or biological properties of those 3 amino acids in that order. More recently, people have used deeper, bigger neural networks to pre-train on language and on proteins, or to learn representations of language or representations of proteins. So one example is it's called an autoregressive language model. And what this does is, given a sequence, you append a start token to it, and from there you want to predict the first item in the sequence. Then given the first item, you predict the next one, and so forth. So at every location, you predict what's going to come next from everything that's come before. Another idea is what's called a masked language model. And here, we start with that sequence, and we randomly corrupt it by covering up with a special mask token some percentage of the amino acids. And we also randomly change some other ones. And there's some we keep the same. We also want to make sure we predict those and recover those. And now we run that through a neural network and try to reconstruct the things we've corrupted. And the idea here is that given everything else around it, we should be able to figure out what should go at a certain location, or the model should be able to figure that out. And once again, this final layer we can use as an embedding or a representation of each amino acid in its context. Right now, most people will implement either autoregressive or masked language models using what are called transformers, which at every location learn how much to aggregate information from the other locations. And we do that over and over again to learn meaningful representations. Now I'm going to focus on the neural autoregressive and masked language models. First, we're going to talk about what those can learn. So first, if you look at the embeddings they learn for the individual amino acids, those recapitulate biophysical properties such as charge, hydrophobicity, aromaticity, polarity, and size. And these results are from a PNAS paper by Alex Reeves and co-authors. In that same paper, they show that if you then take the output embeddings and stick a linear projection on it, you can train that linear projection very quickly to predict whether 2 locations in the protein are in contact. So these embeddings contain structural information. And it turns out that the better the pre-training loss, the better the masked language model pre-training result, the better your ability to predict secondary structure and context. These models also contain evolutionary information. One easy way to check that is that you can show that their pseudo-likelihoods, so the probability the masked language model assigns to a mutation divided by the probability of the wild type correlates fairly well with how well that mutation is tolerated. So you can compare 2 masked language models, ESMB, which is and tape, and then a VAE, which is meant to do this kind of mutational tolerance prediction. And you compare it across a whole bunch of what are called deep mutational screens, deep mutational scans, where people have tried to measure the effect of every mutation and see that these mass language models just trained on a whole bunch of proteins straight from UniProt do pretty well. And you can then use that pseudo likelihood to predict an evolutionary vector field by just comparing for two related proteins whether the mutations are likely to go one way or the other, and then predict directionality between those, and then do it for a whole bunch of proteins in the network. And you can validate this, for example, on flu-A nuclear protein, which has been monitored and sequenced for quite a long time. And you show that the direction you can infer from this vectors field correlates fairly well with the year in which these were sequenced. And this evolutionary information contained in the pretrained model also helps guide your search when you're optimizing for sequences. So for example, Biswas et al. Showed that if you train a model on just 24 examples of GFP mutants and how bright they are, and then try to use those models to find really bright variants of GFP and then make those in the lab, you don't get a very good hit rate of things that are better than wild type. And that makes sense because you only have 24 training examples. And they did this across 5 trials where they changed the 24 training examples. But if first you take your wild type GFP and do a homology search and get a few thousand related sequences and do your mass language model pre-training on that, and then fine-tune that model, your hit rate improves about 2%. And if, in addition, before you pre-train on homologous sequences, you first pre-train on a broad variety of protein sequences, your hit rate goes up to somewhere between 5.5% and 11%, depending on, I think this is a model replicate. And why is that? Well, it turns out that with pre-training, that evolutionary information means that the first principal component of your representation actually already begins to separate the dead sequences, the ones that aren't fluorescent at all, from the ones that do fluoresce. And as you add even just a few training examples, 24 and 96, you can quickly see that your predictions get pretty good. So those are some impressive results just based on treating proteins as sequences of characters and pre-training based on that and fine-tuning your task of interest. However, it turns out that protein sequences are not actually human languages. For example, the most obvious thing is that we have 3D structure here. You go from the primary sequence or structure that falls into secondary structure, such as sheets and helices, and then the 3D structure, the tertiary structure is the 3D structure of one protein sequence, and then something that's combined together to form quaternary structure. There are 20 amino acids, and versus words and subwords in language, there are many more words and subwords than 20 amino acids. And there's also natural divisions between words and subwords, whereas within a protein sequence, it's not immediately obvious what those divisions should be. And to a first approximation, if you go and scrape the internet for examples of language, you can assume that those are almost IID in their generation process, whereas it's definitely not the case for protein sequences. There's 2 confounders here. One is evolution. So all proteins from Earth evolved from common ancestors, so that biases the sampling. And in addition, we haven't collected a uniform sample of all the proteins on Earth. We collect things in humans a lot, in mice, in certain bacteria, but there's a lot of things out in the ocean or inside volcanoes that we might not be able to sequence. And to kind of take advantage of these differences and the different data sources we have, I would propose or suggest that we start training multimodal models that take advantage of this. So we've been talking about pre-training with sequence. We can also use evolutionary information or structure. Or if we're talking about enzymes, if you know what reaction they catalyze, that chemistry could be helpful. These databases also come with a whole bunch of annotations. Go is a set of functional annotations. There's PFAM domains. You might know what thing they bind to. There's other annotations like KEG, etc. And people have put a lot of effort into these annotations, and there's probably a good amount of information contained there. And then for a lot of these proteins, there's also free text. UniProt will link to publications, and sometimes you can see titles or abstracts. So there's also information there that we can take advantage of. Now I'm going to give some examples of people doing these kinds of models, beginning with an example involving sequence and structure. So I've talked about this mass language model just in sequence, but it turns out if you know the structure of the protein you're dealing with, you can do much better in that mass language modeling task. And here's one example of how. I'm going to call this a structure-conditioned mass language model. And so your goal is to predict the mass locations given the structure and the unmasked locations, whereas before we just predicted the mass locations from the unmasked locations. So here's kind of a cartoon, and I've masked that blue token in the middle, and I'm going to get information from its neighbors, both sequence information and structural information. And I'm going to use that to predict the identity of that masked token. And I'm going to take 19,000 examples from Cath, they're duplicated, and you get the structure and the sequence, you mask the sequence like before, and you use those to make node and edge embeddings and feed those into a message passing graph neural network and predict outputs. And I think the first paper to do this was by John Ingraham and co-authors at NeurIPS a few years ago. And if you do this, you can get a perplexity of about 5. And so perplexity is exponential across entropy. Lower is better here. And earlier we talked about what if we train a mass language model on just sequences. If you do this in UniRef 50, there are 42 million sequences. And that sequence-only model in the same test set gives you a perplexity of about 10.86. And that's much worse, even though there's many, many more training examples there. But it turns out that there's still information from the sequence-only pre-training that can be helpful when you have the structure. So if I just take my trained sequence-only model and replace the sequence node embeddings with its outputs, I can drop that perplexity down to 4.65. And of course, there's work to be done here on, can we use this to generate sequence-given structure, or is this a good pre-training regime for fine-tuning or for representation learning? But it seems promising. Another approach for combining sequence and structure is just to use the structure as a target in the pre-training. And this is proposed by Tristan Bepler in a couple of papers now. And the idea here is in this case, we're using LSTM for their mass language modeling. And that's just a standard mass language model task, and that's fine. But then you also take that encoder and make it predict contacts, so predict which locations in the protein are close to each other. And you do another task where you take 2 sequences and try to predict how similar their structures are from this embedding. And if you do this and fine-tune on these same data sets where, once again, these are deep mutational scanning data sets where somebody has tried to get a measurement of how well every single mutation is tolerated in the protein, and they're comparing to a bunch of different methods. So their method is in red, which is a Gaussian process trained on these embeddings that have been trained in sequence and structure. And previous methods are in other colors or other shapes. You can see that for most of these, red is the top, which is good. So combining sequence and structure seems to help here. Now I'm going to give a taste of some other ideas that I find interesting or intriguing recently. So one idea is going back to those Go annotations and other annotations that are in these databases. What if we also combine that with sequence? So in this case, it's a model called Protein Bird by Brandes and co-authors. And on the left side, we have a fairly standard masked language model, but instead of using attention, they're using convolution. But on the right side, what they're doing is they're putting in the annotations, and they're feeding that information into the sequence side, but they're also masking those and reconstructing them. So now, in addition to the sequence information, you also get functional annotation information. Your model has to deal with both. Another one I find interesting, and this one combines chemistry and text, but it doesn't actually use sequence information. So the paper by Crutter and co-authors, and they take the U.S. Patent Office chemistry data set, and they tokenize the smile strings, and they put it through a transformer, and here they pre-train by putting in the reactants and predicting the products, all as tokenized smile strings. So after pre-training on that, what they do is they take in a smaller data set of enzymes and enzymatic reactions, and then descriptions of the enzymes as either just a label of what the enzyme function is or what type of enzyme it is, or sometimes they have whole sentences describing the enzyme. And so once again, they're taking in, this model takes in the substrates, but now it also takes in text information about the enzyme, and then put all of that together and predict the product. And so looking at some results on the test set, looking at top one accuracy, or how many of the top one predictions are the correct product, but also how many of the top one predictions or top two predictions are invalid smile strings. So if you don't use any text, and these are some different pre-training regimes and test regimes, you do okay. If you add in just words that have ACE systems, basically the name of the enzyme, for all of these, you do a little bit better compared to before. And now if you add in full sentences, you improve performance even more. So let's talk briefly about future work here. I've briefly covered some possible sources of information from multimodal models. And I think there's lots of room for creativity there in how we combine information in the model architectures and tasks. It's also important to know exactly how well these pre-training tasks work and downstream tasks. So one way to do that is by developing function prediction benchmark datasets. There are some current protein function ML benchmarks, but they don't discriminate super well between different pre-training regimes. Every reasonable regime does approximately as well as every other. And here it's also important to create challenging and biologically relevant train test splits. Another thing I think is very interesting is uncertainty calibration. So not only do you want to make accurate predictions, but you want to know when your predictions are unreliable. And we need this because a lot of the optimization schemes I described earlier rely on something called Bayesian optimization, which requires well-calibrated uncertainty estimates. And this is especially challenging for combinatorial and high-dimensional sequence inputs like protein sequences. With that, thanks everybody for coming.